My name is Sarah Hennigan, and I began my PhD in the Zoology Department of Trinity College Dublin last September, studying the growth and development of spinal ligaments. My poster is entitled Investigating Movement as a Regulator of Spinal Ligament Development in the Embryonic Chick. We aim to characterize any morphological, mechanical, or molecular differences in spinal ligaments when immobilized, establishing the role of movement in the growth and development of spinal ligaments. We have previously established the chick as an appropriate model to investigate spinal ligament development. The chick is amenable to chemical mobilization, both rigid with no dynamic forces and flaccid with no dynamic or static forces. Details on the mobilization regime we used can be found on the poster. Analysis was primarily focused on the dorsally located supraspinous ligament and the ventrally located anterior longitudinal ligament, both of which run the length of the spine. The location and appearance of these ligaments can be seen in section A. Histological analysis performed includes measuring thickness, cell density, and collagen alignment of these ligaments. The mean thickness of all treatments were below that of their respective control, with the rigid anterior longitudinal ligament being significantly so, as seen in the graphs in section A. In section B, you can see we analyze cell density by staining and counting nuclei in the supraspinous ligament. We found a significant increase in cell density with rigid immobilization, suggesting that immobilization disrupts tissue organization. In section C, we describe our approach to analysis of the alignment of collagen fibers. We are quantifying alignment via polarized light microscopy on Picoceres red stained longitudinal sections. Fibers were quantified according to their degrees of deviation from zero, defined as a horizontal line across the image. The percentage of fibers within 20 degrees of zero have been plotted on this graph. Analysis is still ongoing, however preliminary data show the means of both treatments being below that of the control. We performed uniaxial tensile testing on both the dorsal supraspinous ligament and the ventral anterior longitudinal ligament. Initial and final elastic moduli were calculated, shown in section D. In the supraspinous ligament, we can see that the flaccid treatment results in samples significantly more elastic than the rigid or control samples. However, we found that both treatments were significantly more elastic in the anterior longitudinal ligament compared to the control. This suggests that different ligaments may experience differing impacts of immobilization. These differences between ligaments can also be seen in the molecular analysis we conducted. In section E, you can see the changes in gene expression we quantified via QRT-PCR. We found significant differences in collagen genes, extracellular matrix genes, and the transcription factor MOHAWK. Overall, this research has established that movement does have a role in the correct patterning and development of spinal ligaments. We have found morphological, mechanical, and molecular differences in spinal ligaments under immobilization. We aim to use our research to aid in our understanding of spinal deformations, and our future work will investigate spinal ligaments in the mouse and in scoliotic spines. If you would like to get in contact, my name and email is located at the bottom right of this poster. Thank you very much.